Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is plant growth and development. In the previous lectures, we have discussed already about what is the growth, what is the development, how development occur, how growth takes place, what are the different type of growth, what are the growth rate, all these things, right, and the factors which affect them. That all thing we have discussed. And one more important topic, that is plasticity or heterophilia, also will be discussed in the previous one. And we have already talked that in development there is some extrinsic and intrinsic factor, and we have discussed that in the last slide of this last lecture. We are continuing today out of this the intrinsic factor which synthesized by one cell and goes and show a effect on the another cell, that is intercellular component, that is a plant growth regulator. So we are going to discuss here today mainly about the plant growth regulators. So this plant growth regulators means they are going to regulate. So in regulation, both things can happen. Either it can promote, either it can inhibit. Okay. So we will be going to talk about the plant growth promoter as well as the inhibitor. Okay. So we will discuss that. So here we are going to discuss plant growth regulators mainly. So what are they? So obviously, these plant growth regulators are nothing but a chemical compounds, or we can say they are the small simple molecules of diverse chemical composition. They are chemical compounds. And this chemical help compounds ultimately helping a plant in a growth, or they may be inhibiting some of the activities. So plant growth regulators, if we talk, are variously described as a plant growth substances. They can be defined as a plant growth substances or plant hormone or phytohormone in the literature. Okay. And obviously, they regulate the growth, differentiation, and development by promoting or inhibiting the activities at a very low concentration. So this is the measurement oxynometer which we have discussed already. Okay, so plant growth is an important one. So the PGR can be broadly divided into, they are chemical compounds and they are broadly divided into two groups based on their function in the living body. And these plant growth regulators, they are of two types, that is the growth promoters and the growth inhibitors. So this plant growth regulators, or we can say which are the growth promoters or growth inhibitors, in which growth promoters, if we talk, in which they promote some of the activities like cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, seed formation. Plant growth regulators are called a growth regulators as well. And growth inhibitors, they inhibit some of the activities like dormancy and abscission, like inhibiting activities also they perform. So there are growth inhibitors as well. So the plant growth regulators of other group play an important role in the response to wound and stresses of biotic and abiotic origin. Obviously, they particularly play a role, and some of the healing of wound and stresses like biotic and abiotic origin. There are also in various growth inhibiting activities that the what inhibitors such as a dormancy and abscission, and the plant growth regulator abscisic acid belong to this group obviously. Ethylene is a plant growth hormone or regulator which is gaseous and that could fit either of the group, but it is largely an inhibitor and it has a majority growth inhibiting activity. So type of plant growth regulators, if we talk, which are the chemical substances other than the nutrient produced naturally in plant, and this plant growth regulation may be translocated to another region for regulating one or more physiological activities when present in low concentration. And here five natural plant growth regulators are there. One is auxin, another one is a gibberellin, third one is a cytokinin, fourth one is a ethylene, and the fifth one is abscisic acid. These five types of phytohormone are known, and in this first we are going to discuss auxin, then we will discuss another one. Now in this thing, we remember that this auxin, gibberellin, and cytokinin are the promoters, whereas this ethylene and abscisic acid are the plant growth inhibitor. Okay. So let's discuss auxin in detail. So growth regulator and their chemical composition, before discussing auxin in detail, all we have to compare. So auxin is an indole compound and its Chemical name is indole 3 acetic acid, and indole 3 acetic acid is obtained naturally from the plant. Another one is the cytokinin, and cytokinin is adenine derivative, and this adenine derivative is N6 sulfonyl aminopurine, that is the kinetin. Another one is the abscisic acid, that is a derivative of carotenoid. Then gibberellic acid, which is the terpenes, and the ethylene, which is the gases. Okay, so these five plant hormone are there, and these are their precursor, their chemical actually their chemical composition that we have discussed. We will discuss again in detail. And first, we are going to obviously discuss the auxin, which is a plant growth regulator. Okay, so auxin, it is a plant growth promoter. Okay, so we are going to discuss auxin in detail. So if we talk about auxin, it is a weakly acidic growth hormone. It is weakly acidic growth hormone. Okay, and this auxin have unsaturated ring structure, unsaturated ring structure, and capable. It has a unsaturated ring structure, and this auxin, it is capable of promoting cell elongation, especially of shoot, especially of shoot and shoot segment at a concentration less than 100 parts per million. Okay, so this is obviously weakly acidic. They have unsaturated ring structure, and they mainly has a pronounced effect, or especially on the shoot and shoot segment, it has a cell elongation effect at a concentration which is less than 100 parts per million. And obviously, this concentration of auxin is inhibitory to root. Is inhibitory to roots. And among the growth regulator, auxin were the first to be discovered. Auxin, it is first discovered growth regulator. First discovered growth regulators. Now, however, the discovery of auxin and other type of PGR have been accidental or are accidentally discovered, but these are the particular hormones. Now, in this, if we talk about auxin, and first, if we discuss about the history or discovery of auxin, then dear student, the first discovery was the first scientist to show play a role in a discovery of this auxin, that is a Charles Darwin, and his son Francis Darwin. And during 1880, they carried out this particular experiment. Now, this Charles Darwin and his son Francis Darwin found that unilateral illumination was unilateral illumination of light, unilateral illumination of light was was picked up by coleoptile tip, coleoptile tip of canary grass. That is Phalaris canarensis. So Phalaris canarensis was the scientist for canary grass, was the particular, Phalaris canarensis, the canary grass on which this unilateral means from one side a light is given, and he particularly shown that this particular Phalaris canarensis, a coleoptile tip, pick up this particular illumination. And he observed that the decapitated coleoptile say, coleoptile did not receive sensation, did not receive sensation. Now, how they show? Suppose this is a coleoptile tip. If we give unilateral light, it will show a bending like this. Okay. So bending occur when we give a unilateral light to the this coleoptile. But if we carry out a decapitation like this is the removal of cap. So this decapitation occur, and like this is the decapitated coleoptile tip. Now this show no bending. So if it is not showing the bending, okay, then that is a decapitated coleoptile, which is where cap is removed. So if cap is not there, it is not able to receive the stimulus, and because of this not ability to receive the stimulus, that is not showing the bending, means we can say the decapitated coleoptile cannot receive the sensation. Then he done one more thing. What he did? He covered the coleoptile tip by an opaque tin foil. This is the coleoptile, and this coleoptile tip is covered by opaque tin foil. And when it is covered by opaque tin foil cap, he found that no bending occurred. Means this opaque tin foil cap could not receive the stimulus of light. Means opaque tin foil does not receive stimulus of light. Whereas so this is the thing what he observed. Decapitated does not show the particular what bending. Same way, opaque tin foil covering will not particularly allow covering. But if this cap, this coleoptile is covered by a translucent cap, then it show bending, means bending occurred. Means the particular light stimulus passes through this particular, we can say, the translucent cap of the slit. Now, so what Bison and Jensen proved? So they proved that the light stimulus is picked up by is picked up by canary grass coleoptile tip and and transmitted to subapical part, transmitted to subapical part, which bend which bend in a relation to direction of light. So if this is a coleoptile tip, you can see if bending occurred, bending will be like this. So this bending is the what shown in the subapical part. So this subapical part shows the bending that was proved by this particular Francis Darwin and Charles Darwin. But after this, the second group of scientists who worked over this, that is the Bison and Jensen. Second group of scientists is Bison and Jensen from 1910 to 1913. He worked out this experiment. What they showed? They showed that the light sensation of light stimulus is picked up by coleoptile tip coleoptile tip that could be transmitted that could be transmitted through block of gelatin, but not through mica plate. But not through mica plate. Now, how he done this experiment? Suppose this is the coleoptile tip. Here, mica plate was kept. If mica plate is there, this mica plate do not show bending. It remain like this. So means no bending. But if the gelatin block is kept here, it shows bending. So if gelatin block we keep and bending occur, means the particular sensation or stimulus or the hormone which is getting synthesized, that obviously passes through the gelatin block. Okay, so that is the Bison and Jensen. After this, the third scientist who played a role in the discovery of oxygen, that is Paul. In 1919, Paul replaced what Paul did. Paul replaced the excised tip eccentrically over a stump of coleoptile. Over a stump of coleoptile means if this is the tip, we cut this tip, and on decapitated tip, we are keeping it eccentrically. Then it show bending. So what he observed? He observed greater growth on that side where this particular cap was kept eccentrically, even in dark. Okay, so this was a particular scene during the PAL work. But now we are going to discuss one of the important work, and this is the important one. You should remember this. That is the contribution of the FW vent. This contribution is much important. It is given in NCERT, and according to NCERT, also this is important. So vent, what vent did? First, vent specifically worked on oat coleoptile. We remember. What is oat? That is Avena sativa, a monocot plant. So he taken a oat coleoptile and removed a cap. So first what he performed? Decapitation. After decapitation, he placed this tip on agar block. And on agar block, this will be like this. So this passes to the particular agar block. And on our agar block, this particular, you can say, the substance which is synthesized in the presence of light in the tip here get transported to this agar block. Then what he did, on decapitated tip of oat coleoptile, he placed this agar block. And when he placed this agar block, he showed that bending occurred. So he taken the nutrient which is getting synthesized to the agar block, then placed the agar block on the decapitated tip and bending observed. So when collected this growth stimulating substance in agar block, means they can isolate through this agar block. So through by using the agar block, he isolated this particular growth stimulating substance. So next, from agar block, he isolated growth stimulating substance. So growth stimulating substance was isolated. So he discovered that The hormone travel basipitally. He discovered that hormone travel basipitally. That is from tip or apex, from tip or apex towards base. Okay. So from apex towards base, the transport occurs. So agar block containing the chemical cause the bending of a decapitated coleoptile according to its concentration. So he discovered this particular. So hormone travel basically from tip towards the back. So we can say that this growth substance later on, the growth promoting substance was named by him, that is FW went as oxime. So who isolated this oxime from the plant or oat coleoptile? FW went. Who given a name to the particular this hormone as an oxime? So that is also a FW went. And this oxime means simply to grow. Okay. Now after this fourth, the fifth group of scientists which was worked. So FW went contribution is important according to SCRT, that you should remember. Next, Kogel and Hagen Smith also worked on this. And they isolated three chemicals. They isolated three chemicals from human urine. And they are named as oxygen A, oxygen B, and heterooxygen. Okay. So they named this three chemical as a oxygen A, oxygen B, and heterooxygen. But Kogel et al. Found that heterooxygen is the real plant oxygen. Is the real plant oxygen. And chemically, it is indole 3 acetic acid or IAA. Okay, and it is known that it is also present. It is also present in urine of human being suffering from pellagra. And pellagra is a disease caused by deficiency of niacin or nicotinic acid. Okay, so this is the thing which is observed. And obviously, the real oxygen is indole 3 acetic acid. This was discovered by Fogel et al. Okay, so this is all about the what we can the discovery. Now, next thing which we are going to talk about, that is the important thing is that the occurrence of oxygen. That is known that oxygen is synthesized in pseudopoiesis. It is synthesized in pseudopoiesis, leaf primordia, and developing cysts. Now, next, what is the we can say precursor? Precursor is a chemical compound from which this oxygen is getting synthesized. So, precursor of oxygen is tryptophan, and tryptophan is a one type of amino acid. So, oxygen is a particular hormone which is of a amino acid derivative. And obviously, we have discussed that oxygen movement is polar. Obviously, oxygen movement is polar and basal as we have discussed from apex to the base. And one important point is that oxygen helps in elongation of both root and shoots. However, the optimum for the two is quite different. The oxygen for elongation of the root and shoot, the different concentration play a role in that. Okay. So tryptophan is oxygen precursor. That is one of the important points that we should remember this. So we have understood about oxygen. So generally produced by the growth apices of the stem and root from where they migrate to the region of the action that we discussed. Next, this is obviously apice of the stem. This is the apice of the root. Oxygen like indole acetic acid and indole butyric acid have been isolated from the plant, so they are natural oxygen. There are two types of oxygen: natural oxygen and artificial oxygen or synthetic oxygen. So indole acetic acid and indole butyric acid are the natural oxygen. Okay, this is the structure of indole 3 acetic acid, and this is a indole butyric acid. Actually, structures are not of importance for us. This is for just for information. So these are the natural oxygens. Now there are some artificial or synthetic oxygens also, like NaA, there is naphthalene acetic acid. Then 2,4-D dichloro, 2,4-D, that is the 2,4-dichlorophenoxyacetic acid. These are the synthetic oxygens. And apart from this, if we talk here, 2,4-D, one more is a synthetic oxygen, that is a 2,4,5-T. That is 2,4,5 trichlorophenoxyacetic acid. Trichlorophenoxyacetic acid is found here. Naphthalene acetic acid also, particularly we can say synthetic oxygen. But One more important point here we have to remember that indole butyric acid indole butyric acid is both natural and artificial oxygen because it is obtained in plant naturally also, and it can be synthesized in laboratory as well artificially. Okay, so it is a particularly we can say the natural as well as artificial oxygen. So this is what about introductory part of oxygen. Now oxygen of two type based on natural or artificial that we have discussed, but again oxygen are of two type, that is free oxygen and bound oxygen. And bound oxygen. Free oxygen and bound oxygen. Free oxygen is the oxygen which can be extracted, which can be extracted easily. Okay, and bound oxygen, if we talk about free oxygen that can be extracted easily, and the oxygen which cannot be extracted easily, that is bound only. It cannot be extracted easily because they are not free in the cell. They are bound to any chemical compound. Here they are free in cell, so they can be extracted easily. So if we talk about example of bound oxygen, then indole acetic acid, aspartic acid, indole butyric acid, alanine, okay, indole acetic acid, glycoprotein. So they can be bound to such chemical compound and known as bound oxygen. Actually, bound oxygen is hormonally inactive. It is considered that. So these are hormonally inactive. So they are meant for storage purpose. Okay. So they are meant for storage. Whereas if you talk about a free oxygen, which cannot be, which can be extracted easily, that is the active. Okay, so this is the two type of particularly we can say oxygen, free oxygen, the bound oxygen. Next, after this, there are some inhibitors of oxygen as well, and which are known as antioxidant, which are known as antioxidant. After this, we will talk about the functions. So antioxidants, if we talk, they inhibit a action of oxygen. So they are known as antioxidants. And if we talk about the examples of antioxidants, the first example is PCIB, that is para chlorophenoxy isobutyric acid. Para chlorophenoxy isobutyric acid. Whereas the TIBA is 235 triiodobenzoic acid. Triiodobenzoic acid. That also act as an antioxidant. And this antioxidants, if we talk, they block the transport of oxygen. And such a way they inhibit the activity of oxygen. Okay. Now, after this thing, antioxidants, now we are going to talk about the functions. So all these oxygens which I have discussed till now, discovery, the artificial, natural, bound, free oxygens, these all oxygens have been used extensively in agriculture and horticulture. So this is the direct question which you can get from NCERT, that which hormone is used extensively in the agriculture and horticulture practices. So you should answer that it is a oxygen. They also have to initiate a rooting in stem cutting. They know to particularly cause a rooting. So here we are going to talk about what functions of oxygens from here onwards. Okay, so first function is their role in the particularly agriculture thing. The second important function that they help in a initiate rooting. They develop a root, initiate a root on the stem. It means if we have a cut piece of stem, we can induce the rooting by applying this oxygen. And this application widely used in plant propagation technique. So this is the cut segment, obviously, that can be grown in a dilute oxygen in the glass or beaker, then can be sown. So new roots can be developed easily. So initiative oxygen, initiative rooting. Apart from this, one more thing is that it promotes flowering. Promote flowering. So it promotes flowering specifically in case of, we can say, pineapple. Okay. So oxygen promotes flowering. That we should remember. They also help to prevent a fruit and leaf drop, or means we can say abscission in early stages, but also promote the abscission of older matured leaves and fruit. So that is one of the important role of oxygen, that it delay abscission of young leaves and fruit, but it promote abscission of the matured leaves and fruit by the formation of abscission layer. And this abscission layer cuts off the nutrients and water supply. Because of this, the older matured leaves and fruit falls, and this is known as abscission. Okay, so this is the abscission. And this is, you can see, abscission layer, a yellow layer which developed, that is on the abscission layer. So we can write what? Oxygen promote abscission in older plant part and prevent abscission in younger plant part. Okay, so this is the one of the important thing. And you can see this is the abscission layer which develop here. So it cuts off the nutrient supply. So it's follow. Next, in most higher plant, the growing apical bud inhibit the growth of lateral axillary bud, and the phenomena is called as apical dominance. So next function of the oxygen is what? Apical dominance. So what is apical dominance? Till the apex is present or apical bud is present, it will inhibit the growth of lateral or axillary bud. Now the question is how and why. So what happens when the apical bud is present? The nutrient get transferred to the apical bud, and the flow of nutrient towards the lateral bud stops. Because of this, the growth of lateral bud get inhibited. Okay, so this happens in the apical dominance. So here, this is the apical bud and this is the lateral bud. Lateral bud is not growing in the presence of apical bud. So when the apical bud is removed, the lateral bud will sprout and will produce a dense bushy growth. So the phenomena is widely used in tree plucking and hedge making. Okay. So removal of shoot tip, that is known as decapitation, usually result in the growth of the lateral bud. Means removal of apical dominance can be done by the removing the shoot tip. And lateral bud or lateral shoot will start growing. So this is widely applied in the tea plantation and hedge making. So in tea plantation also, if we cut the tip, the branches will grow fast and the bushy growth will be observed, which is important for the in the tea plantation. Okay. So apical bud inhibit the growth of the lateral bud by releasing the auxin. It is confirmed that planting the cut ends of the decapitated shoot by a paste of auxin, the lateral bud remain inhibited. Means if we cut the particular apical bud and if we put the paste of auxin on the tip, then also lateral bud will not go. So lateral bud remain inhibited if the apical bud is present. Okay. So this is the one of the important thing, apical dominance function of this auxin. Then auxin also induce a parthenocarpy. Parthenocarpy is a development of seedless fruit. Okay, parthenocarpy is a what? Parthenocarpy develop seedless fruit. So seedless fruit are known as parthenocarpic fruits. So parthenocarpy is also particularly one of the important thing induced by the auxin. Then auxin also control the xylem differentiation and help in the cell division. It controls the xylem differentiation. And also control the division. So in xylem differentiation, the auxin is required in specifically tissue culture. The development of callus or mass of undifferentiated cell is promoted by auxin. De-differentiation of callus offers the presence of both auxin and cytokine as well. So this particularly promotes. Then cell division, auxin known to promote a division or a cell division in cells of vascular cambium. Okay, so in cells of vascular cambium, it is going to promote particularly the cell division. Next, auxin are also used as a herbicide or weedicides. They are also used as herbicides or we can say weedicides. So weedicides are the what? Obviously, they are going to remove the weeds. So here, 2,4-D, that is a 2,4-dichlorophenolic acid that is widely used to kill a dicot weeds and does not affect the mature monocot plants. So it removes the dicot weeds from the monocot field. So obviously, the lawn are the one type of what monocot. Okay, so used to prepare a weed-free lawns. Okay, so weed-free lawns can be particularly created by gardeners by using this 2,4-D. So this can be weedicide. Why here weedicide is acting? Because weedicide or auxin here acting as a chemical which is killing the weeds growing in the field. So application of 2,4-D and 2,4,5-T is also used as a weedicide that remove the broad-leaved weeds. We remember this dicot weeds is the broad-leaved weeds. So we can write 2,4-D and 2,4,5-T remove broad-leaved weeds. That is dicot weeds in cereal crop. In cereal crops. Okay. And lawns because they do not affect a mature monocot and they affect the what particularly dicot. So these are the some important functions. Apart from this, there are several more functions also. It is known that auxin stimulate respiration. Auxin stimulate respiration most probably by increasing availability of by increasing availability of respiratory substrate. So as the respiratory substrates become more available, the respiration can be more. Next, one more function is that oxygen enhance metabolism due to metabolism of plant resource. Apart from this, oxygen also increases storage of solute. Oxygen increase storage of solute in cells. Then it is known that it increases cell enlargement, which is the fundamental activity of oxygen. So cell enlargement is induced or increased by the oxygen. How it does? So it is known that oxygen causes solubilization of carbohydrates, loosening of cell wall microfibrils, synthesis of more wall material, increased membrane permeability, increased membrane permeability, increased membrane permeability and respiration. By carrying out all this event, it causes a cell enlargement that lead to cell enlargement. So if we show this properly in a diagrammatic way, which make us to understand it proper way, suppose indole acetic acid is there. What it does? It carry out a synthesis of amylase enzyme. What amylase will do? Amylase act on the starch. Now starch will broken up into the sugars. This starch also get converted into sugars. Now what will happen apart from this? What another thing that do? That indole acetic acid increase membrane permeability. So if it increases membrane permeability, what happens? The sugar will enter the cell along with salts. And as it enters the cell along with the salt, the osmotic pressure, that is OP, get increased. So this thing happen. Apart from this, what it does? It carry out loosening of cell wall microfibrils. So that will lead to the more cell wall material synthesis. Apart from this, the sugars that carry out increase in respiration, the sugars get available, so increase the respiration. As the respiration increases, there is an increased anabolic activity, and that increased synthesis of cell wall material also will take place, so more cell wall material will be synthesized. and that thing happens along with that osmotic pressure increases. So that lead to the what? Endoosmosis. As the endoosmosis will take place and more cell wall material take place, cell elongation occur. So this way oxygen affects so many activities and carry out the cell elongation or the enlargement. Apart from this, as we have discussed, it controls xylem differentiation, promote the cell division, promote root initiation at a concentration which is inhibitory to the growth of intact root. Apical dominance we have discussed, abscission we have discussed. Next thing which we are going to discuss, that is the tropic movement. So indole acetic acid or oxygen produce tropical plant response, like phototropism and geotropism. Phototropism and geotropism. Apart from this, we have discussed about the parthenocarpy. Apart from this, it is known that higher concentration of oxygen, as we have discussed, higher concentration of oxygen have an inhibitory effect. How? So higher concentration of oxygen or indole acetic acid induces synthesis of oxygen, synthesis of ethylene, sorry. So high concentration of oxygen increases synthesis of ethylene, and we know that it is a gaseous hormone and it has an inhibitory effect. Apart from this, oxygen cause femaleness. So oxygen cause femaleness, one of the important point in some plants. So it has a feminizing effect in some of the plants. So if somebody asks, oxygen promote which type of flower formation? So it is a female flower formation. Okay. Then we have discussed about that it causes rooting on the stem cutting plant. So obviously rooting is induced by the stem cutting, and in that IBA, indole butyric acid, alanine, that is bound oxygen, and naphthalene acetic acid are used. Then flowering we have discussed in case of pineapple, it induces in lychee as well. So it is known that naphthalic acetic acid and 2,4-D that induce flowering in lychee and pineapple. Okay. Then indole butyric acid specifically that enhance sweetening of food. So fruit sweetening is also caused by this particular oxygen. Then naphthalene acetic acid, naphthalene acetamide, that is NAAM. This NAAM stands for naphthalene acetamide, which prevent lodging or falling of crops during windy season. Okay, so these are the some of the effects which is particularly caused by this auxin. If we write one more important point with respect to auxin, then we remember that it is known that application of naphthalene acetic acid increases number of dwarf shoots as well as number of root. Okay, so that is particularly caused by the what? Auxin. Now, last we are going to talk about the bioassay of auxin. And if we talk about the bioassay of auxin, first we have to understand this bioassay are meant for what. So if you talk about the auxin, it is important for the testing of a biological activity like a growth response of a substance by employing a living material like plant or plant part. Auxin bioassay is a quantitative test. This bioassay of auxin are the quantitative test, and it measures the concentration of auxin to produce an effect and the amount of it. Means whatever the effect, whatever the function we have discussed that is caused by this auxin, at what particular concentration they are showing this particular type of effect, that can be understood by this particular bioassay. And two important known bioassay are there. One is Avena curvature test. Now this Avena curvature test is the important bioassay. Two times in Neet it was asked. Okay, in this test is based on the experiment of the band. And this bioassay showed that 10-degree curvature is produced by a auxiliary concentration of 150 microgram liter at 25 degrees. Okay, so this way we can particularly identify the concentration at which such way how much bending can be observed. And the second bioassay is the root growth inhibition test. So this will help us to understand at which concentration the root growth inhibition occur. Okay, so this type of bioassay were performed. So they ultimately identifying the concentration and which the how much degree curvature is obtained or bending occurs, that is through the Avena curvature test, and through root growth inhibition test at what concentration root growth get inhibited. Okay, so this is the what particularly we can say about the bioassay. Much more detail about bioassay no need to remember because they ask direct question which of the following is the bioassay for the exam. Neet about Avena curvature test two times in Neet the same question was asked. One time they asked that which bioassay is for the auxin, then we have to select Avena curvature test. And in the next year, same question as Avena curvature test is the bioassay of, then it is of auxin. Such type of easy question can be asked, so you should be able to answer that particular question. So, okay, so I hope you understood this auxin. That is all about the what auxin in detail. We have discussed about the discovery, the type of auxin, based on the function, based on the occurrence. Then we have discussed about the what particularly how it can be isolated. Okay, then their particular inhibitors we have discussed, then their function we have discussed, and the bioassay. That all things we have discussed till now. I hope you understood that. Thank you everyone.